enough. All right. All right. Question number 1. Does the amount of LDLR mRNA increase or decrease in a cell with low cholesterol? Draw the steps that lead to the increase or decrease. Well, I believe that the amount of LDLR mRNA will increase in a cell with low cholesterol. And so I'm pretty sure the steps for this is basically like the SREBP pathway thingy. So for that, we got our 3 circles that are like touching. Here we have our SREBP. And then we have our SCAP. And then we have our insigs. Yeah. And so these puppies initially start out in the ER. And I'm pretty sure it's one of these two. I think it's a SCAP. When SCAP senses the low cholesterol, it'll take the SCAP and the SREBP down to the Golgi. The instincts, I'm pretty sure, just either stay in the ER or like float off into the abyss. And so the SREBP and SCAP go to the Golgi, and this is where they're cleaved. I'm pretty sure that C in SCAP stands for cleave or cleavage or whatever it is. So it goes to Golgi where it gets cleaved. And for that we have our N terminal and then C terminal. And it gets cleaved into 3 different fragments. Uno, dos, tres. And this first fragment, the one closest to the N terminal, this gets to go down to the nucleus. So like the SREBP N goes to the nucleus. And in the nucleus we have a nice little promoter. This is our LDLR promoter. So we got our 0 plus 1. And we have our ta-da box. Ta-da! Where Paul 2 hangs out. And then, so for this promoter, this SREBP-N will bind to SRE1, or SRE, whatever it is. So SREBP, that N, that first fragment, binds to SRE1, activating the promoter. And so I believe that causes transcription to occur. Yeah, so if transcription's occurring, that would cause an increase in the amount of LDLR mRNA. Yeah, not a decrease. Pretty sure if this bad layer right here, this SREBP-N, did not bind to the SRE, that would not make the promoter active, which would like cause a decrease because it wouldn't be able to produce the LDLR mRNA. Yeah. Okay. Question 2. Set of cells with low cholesterol and a set of cells with low cholesterol that's treated with MK2206. Would the 2 sets have different amounts of LDLR protein? Explain. So I'm pretty sure the answer to this is yes. Yes, they obviously would have different amounts of LDLR protein. So the set of cells with low cholesterol that's untreated with the MK2206, that I'm pretty sure would just do like this normal pathway. Yeah. So I'm pretty sure like it would just, like the SCAP or the SREBP, whichever one it is, that senses the low cholesterol and like gets this whole thing going. It would get the pathway going. This would bind, increase in transcription, which would then get translated into protein, because there's nothing like stopping that from happening. So that would just be like a normal pathway of transcripted into mRNA, translated into protein for that part. Yeah. But that set of cells with the low cholesterol that's treated with the MK2206 would be a little different. So I'm pretty sure one of like the little jobs of MK2206 is to reduce translation. And so although it has low cholesterol, it would go through like this pathway basically, get transcripted into mRNA. But because it's treated with the MK2206, it would be, well, I don't think it could. It would just like reduce the translation levels. So the mRNA, like it would be, the levels that would be produced would be pretty high, but it wouldn't, like the levels would not be the same for the protein because there would be reduced translation. So transcription would be normal to high, but the protein, the translated, or the chip, that from mRNA to protein, the translation that would occur would be reduced due to the MK2206. So those levels would be lower. Yeah. So the untreated would have like normal to high levels of protein, of alveolar protein, and then the treated would have like low. Yeah, they would have low amounts of protein because the mRNA that's produced can't get translated into protein due to the MK2206 that it's treated with. Hell yeah.